Epstein-Barr virus is a member of herpes viridae family. This virus is classified into two types based on MNAT2 and MNAT3 family genes. Type 1 has a worldwide distribution, and type 2 is more prevalent in Africa and Papua New Guinea. EBV is the causative agent of infections mononucleosis and is related with malignant disease. The prevalence of EBV-related malignancies and proportion of EBV-associated case and viral type circulation vary among geographical regions, and this is the way many scientists are looking for molecular markers associated to malignancy. Given next-generation sequences, more than 800 worldwide complete genomes were sequenced. However, South American EBV isolates are unrepresented. Our aim was to characterize polymorphisms in the entire genome of EBV isolates from our region and compare them with sequences from different geographies. 15 isolates from Argentinian pediatric patients were sequenced by next-generation sequences using EBV custom target enrichment proofs. To compare the variability in the context of EBV sequences from other geographies, 200 FASTQ files were downloaded from NCBI database. All the FASTQ files were analyzed with our custom-built mapping-based bioinformatic pipeline. After the preprocessing steps, reads were mapped to both EBV type reference genome. We construct BCF files and use the information contained in these files to predict EBV type. Consensus sequences were obtained from BCF files. Repetitive region and uncovered region were masked. The alignment was generated using math. Phylogenetic analysis was performed using IQ3 with maximum likelihood algorithms. According to our custom pipeline, in the viral typing graph, we can see when I map to the incorrect referent, I expect to discover more variants than if I map to the correct reference. In this case, both genes are necessary to infer viral type and tax recombinant sequences. 194 samples were classified as EBV1. 20 samples were EBV2 and 4 samples were recombinant between EBV1 and EBV2. Those recombinant sequences were cut off from further analysis. Regarding Argentinian isolates, 9 were EBV1 and 6 EBV2. The phylogenetic reconstruction clearly defined two clades which correspond to EBV1 and EBV2. In the EBV1 group, geographical structure was only detected for the South Asian sequences and Indonesia and Papua New Guinea sequences. Argentinian sequences segregated together with African and European isolates, whereas only 3 genomes clustered with Asian clades. Altogether, the mean number of variants for the Asian genomes, which include China, India, Papua New Guinea, and Indonesia, was greater than those for other geographies. On the other hand, the Argentinian sequences were the most heterogeneous group in terms of amount of variants. Moreover, variants in the Asian group were located in different genomic positions with regard to the reference. As concluding remarks, we designed a custom pipeline that allowed us to successfully analyze the first complete EBV genomes in Argentina. In our Argentinian series, EBV2 was more prevalent than previously described. Asian isolates present more variants and in different positions when compared to the reference sequence. it could be advisable to establish new reference for different geographic regions. We expanded data on EBV complete genomes from Argentina and inferred their closer phylogenetic relation with African and European genomes.